Inside every cell, protein synthesis is essential for life. Proteins build structures, transport substances, send signals, and protect the body from disease. Take insulin, for example, a protein hormone produced in the pancreas. It helps regulate blood glucose levels. Without it, glucose can't enter cells, leading to diabetes.
Another vital protein is hemoglobin, found in red blood cells. It binds to oxygen in the lungs and carries it to body tissues. Without hemoglobin, cells would be starved of oxygen.
Protein synthesis allows cells to make proteins like insulin and hemoglobin, molecules that are critical for survival. Protein synthesis starts in the nucleus, where DNA is stored. DNA is a double helix structure made of long chains of nucleotides.
Each nucleotide contains a phosphate, a sugar called deoxyribose, and one of the 4 nitrogenous bases: adenine, thymine, cytosine, or guanine.
A gene is a specific segment of DNA that holds the instructions for building one protein. When a protein is needed, a signal activates a specific gene. The enzyme helicase unwinds and separates the DNA strands, exposing the template strand.
The enzyme RNA polymerase reads the template strand and builds a complementary strand of messenger RNA or mRNA.
In this process, adenine on DNA pairs with uracil on RNA. Thymine on DNA pairs with adenine on RNA, cytosine pairs with guanine, and guanine pairs with cytosine. This creates an mRNA copy of the gene that carries the code for building a protein.
Once the mRNA is complete, it exits the nucleus through a nuclear pore and enters the cytoplasm, where the next stage, translation, begins.
In the cytoplasm, a ribosome attaches to the mRNA at a specific start codon, adenine, uracil, guanine, which signals the start of translation and codes for methionine, the first amino acid in every new protein. Transfer RNA or tRNA molecules carry specific amino acids to the ribosome. Each tRNA has an anticodon that matches a codon on mRNA.
As the ribosome reads each codon, a group of 3 bases, tRNA brings the matching amino acid.
The ribosome joins the amino acids with peptide bonds, forming a polypeptide chain which will fold into a functional protein. The completed protein detaches from the ribosome and begins folding into its final shape so it can function in the cell.
Every molecule of insulin is made this way, starting with a gene inside pancreatic cells. Hemoglobin 2, made by red blood cells, begins with a DNA message transcribed into mRNA.
Without protein synthesis, none of these crucial molecules could exist. From DNA to protein, this process is called gene expression, and it's happening in your cells every second of every day.